Welcome back. This is my final presentation in this series on reproductive PRRS. I am John Harding, Professor of Swine Production Medicine at the Western College of Veterinary Medicine, University of Saskatchewan. In this presentation, I will discuss the results of a genome-wide association study that was designed to identify genomic markers associated with PRRS severity in fetuses. As you are no doubt aware, genome-wide association studies require large numbers of animals, so the pregnant gilt model, undertaken in 2012 at the University of Saskatchewan, provided this unique opportunity. It remains the only reproductive PRRS GWAS performed on animals challenged in a controlled environment and provides fundamental results that can be validated in future experimental studies or in the field. I would again like to acknowledge my collaborators who deserve much credit for helping to unravel the mysteries of reproductive PRRS. In particular, Tianfu Yang, a PhD candidate of Dr. Graham Plastos at the University of Alberta, who completed this analysis and published the results in scientific reports. The objective of this lecture is for you to understand the potential relationships between host genomic markers and the severity of reproductive PRRS. By the end of the presentation, you should also understand how selection for PRRS resilience might enhance control programs in the future and some of the challenges that must be overcome when studying genomic resilience for reproductive PRRS. The pregnant gilt model is the largest PRRS experimental challenge study performed to date and provided the biological samples and data required to conduct this genome-wide association study. Because our main interest was studying PRRS resilience in commercial female lines, purebred landrace gilts bred to York boars, thereby producing F1 progeny, were used. Our first experiment used 15 gilts and was designed to set up the animal model and select the appropriate PRRS strain based on its reproductive virulence. The second experiment was designed to investigate phenotypic and genomic factors associated with PRRS resilience. Pregnant gilts were inoculated at 85 days of gestation and were terminated 21 days later at gestation day 106. We did not carry the gilts to term because collecting samples from each fetus in situ and understanding its relationships to its neighbor were very important. Of the 114 PRRS inoculated gilts started, 2 aborted and 1 died, so 111 gilts and their progeny completed the experiment. After termination, the preservation status of all fetuses was assessed. This slide shows the breakdown of fetal preservation by inoculation group. Viable fetuses were normal in appearance with whitish skin and engorged umbilical cord. Meconium-stained fetuses were also alive at termination, but covered with pasty yellowish-brown amniotic fluid. Decomposed and autolyzed fetuses were dead, with variable signs of decay. The skin of decomposed fetuses was generally white, whereas autolyzed fetuses were mostly brown-skinned and the carcasses in advanced stages of decay. For this GWAS, the 2 outcome variables of interest were fetal preservation and viral load. This was based on the assumption that for a fetus to be resilient to PRRS, it would be more likely to tolerate infection than to completely resist infection. Thus, an infected viable fetus with low viral load was considered a positive outcome, in contrast to a dead fetus or a fetus with high viral load that would be congenitally infected at birth. This slide shows fetuses, represented by the colored circles, in their respective locations within the uterus. The colors are representative of the fetal preservation status: viable in dark green, meconium-stained in light green, decomposed in yellow, and autolyzed in orange. The log 10 per milligram PRRS viral load is the number in parentheses under each fetus. It is not hard to see that there is a lot of variation in both fetal preservation and viral load within and between litters. Some of this can be explained by the viral load at the maternal-fetal interface, but we are convinced that the responses in the fetus are an equally important piece of the puzzle. DNA was extracted from 993 out of the roughly 1,400 PRRS-infected fetuses available for this GWAS. We were not able to extract DNA of sufficient quality from any of the autolyzed fetuses, so they were excluded. Fetuses were genotyped using the Illumina porcine SNP60 bead chip, which yielded over 45,000 SNPs after quality control. The presence of these SNPs was assessed in relation to 4 outcome measures of interest: fetal viability, fetal death, viral load in maternal-fetal interface, and viral load in fetal thymus. We'll discuss each of these in more detail in the upcoming slides. Firstly, these histograms show the distribution in viral load across the 933 PRRS-infected fetuses included in the GWAS. On the histogram on left, about 30% of the samples of MFI were negative, i.e. with 0 viral load, and the remainder had viral load between 0 and 8 log 10 per milligram. Values were similar in fetal thymus, shown on the right, except that about 50% of the samples tested negative. This Manhattan plot identifies the SNPs associated with viral load in the maternal-fetal interface, and in this case, none are significant at p less than 0.05. This is not unexpected because most of the endometrium and fetal placenta are infected with 1 to 2 days of the dam being infected and well before termination at day 21 in this experiment. By contrast, 3 SNPs, one on each of chromosomes 1, 12, and 14, were associated with viral load in fetal thymus, together accounting for just over 13% of the total genomic variance in viral load. Recall that at each chromosomal location marked by these SNPs, a fetus has 2 alleles, one contributed by the dam and one contributed from the sire. These plots show the effect of these SNPs on thymic viral load. Look at the plot on the far left for the SNP on chromosome 1. It shows viral load on the y-axis for homozygous BB fetuses, homozygous AA fetuses, and heterozygous AB fetuses. You will note that the A allele is favorable and the benefits of multiple A alleles is additive. For example, compared to BB fetuses, viral load decreases about 1/2 log in fetuses with 1 A allele and an additional 0.5 log for fetuses that have 2 A alleles. A similar trend is seen with the SNP on chromosome 14 on the far right plot. The A allele is beneficial and additive, and the benefit of having 2 A alleles is about 1 log 10 of virus per milligram of tissue. The effect is slightly different with the SNP on chromosome 12 seen in the middle plot. In this case, the B allele is favorable but is recessive. Only fetuses with 2 B alleles benefit from lower viral load. Well, we all know that nature sometimes throws curveballs, so we wondered how the lucky fetuses that had mostly favorable alleles would compare to the unlucky fetuses which had mainly unfavorable alleles. So we assessed viral load based on the number of favorable alleles each fetus had. The 3 histograms shown in this slide show the distribution of viral load in thymus amongst fetuses with 1, 3, or 5 favorable alleles in the top, middle, and bottom plots respectively. You can clearly see that more favorable alleles is associated with a higher proportion of fetuses with low viral load. For example, look at the bottom plot. Although very few fetuses had 5 favorable alleles, 16 in total, 60% of these had no detectable virus in thymus. In the middle plot, showing 3 favorable alleles, the percent of fetuses that tested PRRS virus negative decreased to 43%. The top plot shows fetuses with only one favorable allele, and you can see that only 20% of fetuses tested PCR negative in fetal thymus, and many had high viral load as seen by their position on the right side of the plot. Thus, more favorable alleles is associated with decreased viral load. Now let us turn our attention to fetal viability performed by contrasting viable fetuses to meconium and decomposed fetuses. For this outcome trait, we discovered one SNP on chromosome 7 that explained an impressive 34.6% of the total genomic variance. By contrast, 15 SNPs were significantly associated with fetal death. In this comparison, decomposed fetuses were contrasted to viable and meconium-stained fetuses. Together, these 15 SNPs explained 33.5% of the total genomic variance, with 2/3 of this explained by the 7 most significant SNPs with p-values less than 0.001. These SNPs are seen above the upper dashed line on the Manhattan plot. When we compared fetuses based on the number of favorable alleles in their genome, we found that as the number of favorable alleles increased, i.e. moving from left to right on this bar plot, the proportion of decomposed fetuses represented in black decreased, while the proportion of viable fetuses represented in green increased. In the bar plot, the width of the bars represent the number of fetuses within each favorable allele group, and although the data is sparse at the extreme ends of the y-axis, the positive effects of favorable alleles is pretty clear in the central portion. Although this plot is specific to the 7 most significant SNPs, the same trend was seen when we assessed all 15 SNPs shown in the previous slide. One of the limitations of genome-wide association studies is that we don't really know if the significant SNPs we identify in the analyses or in the genomic regions they are within have any biologic function that is relevant for the outcome of interest, in our case, fetal viral load, viability, or death. The data on this slide helps to alleviate that concern. It shows that some of the SNPs identified in the GWAS were in close proximity to genomic regions related to innate or adaptive immunity, other pathogens such as Salmonella, or production outcomes such as mummies. The SNPs associated with fetal death on the right are of particular interest because genomic regions in close proximity include genes related to adaptive immunity, such as interferon gamma and CD4 positive T helper cells, innate immunity, such as TLR2, and other pathogens, such as Salmonella. In summary, this study identified SNPs in 21 candidate regions that were associated with 3 biologically relevant traits of reproductive PRRS. Together, they explained a sizable percentage of the total genomic variance associated with viral load, fetal death, and fetal viability. Some of the SNPs are in close proximity to genomic regions related to immunity and health. But even though these are quite impressive results, they are based on one challenge experiment, one PRRS2 strain, and one breed of gilts. There is more work to be done to demonstrate the external validity of these results. Therefore, before commercial adoption, we need to confirm these results with additional experiments, different PRRS strains and breeds, and in the field. Some of this work is underway, so hopefully in the next few years we'll understand how best to incorporate reproductive PRRS resilience biomarkers into selection and breeding programs, thereby providing an additional tool to improve reproductive PRRS control. That brings this session to an end. I would like to thank our collaborators and funding agencies, particularly Genome Canada, Genome Prairie, and the Saskatchewan Ministry of Agriculture and Food. Additional details can be found in the following references. Thank you for listening.